The overall goal of the following experiment is to probe changes in network function of the brain areas affected by an experimental model of mild traumatic brain injury. Experiments begin by administering a lateral fluid percussion injury to a mouse, which mimics both the pathology and symptomology of human mild traumatic brain injury. As a second step, a hippocampal-dependent behavior, contextual conditioned fear response, is used to assess brain injury-induced cognitive impairment. Next, extracellular and intracellular recordings, along with voltage-sensitive dyes, are used to determine the regionally specific changes in synaptic balance between excitatory and inhibitory synaptic transmission in the region of interest, the hippocampus. The results show subregion-specific changes in net synaptic efficacy in the hippocampus based on differences between the control and brain-injured mice across the 3 recording techniques described.
The main advantage of this approach is the ability to discern the effects of both the pathological state and the potential therapeutics on multiple physiological scales, allowing a more coherent picture of the circuit deficits.
This method can help answer key questions about traumatic brain injury, such as specifically which neurons are most susceptible to injury and therefore making the largest contribution to hippocampal dysfunction after injury.
The implications of our approach extend toward the treatment of traumatic brain injury, as it allows us to delineate first the underlying mechanisms and then develop potential therapies to reinstate adroit